and the students. I would like to express my gratitude to Professor Mahapatra and also to Dr. Gunadidhi, who has invited me to come for this conference to attend. At first I thought of just attending with my scholars, but she said, no, you have to give an invited talk. So thank you. So it was just 4 or 5 days before when I checked my mail, then I got this. Well, before I would start the talk on this topic, I would like to give a little bit history and gratitude to the Department of Utkal University, Odisha, which has, I would say, motivated me to take up this line of research. It dates back to the year 1990s, early 1990s, when I was just a young faculty and a scholar. I was sent by my teacher to attend the training program which was held in the Department of Zoology Utkal University, and it was there that Professor Mohanty Hejmati that time was the head of the department. They run the training for nearly 2 weeks, and I met Professor Datta that time. Then we keep in touch, and then all of us with department, I always have a contact with the Department of Zoology Utkal University, which has really motivated me to take up this line of research. So the gratitude goes to the Department of Zoology Utkal University. Then later with Professor Mahapatra, then Dr. Gunadidhi, we were together, keep in touch. And I hope that this will be a continuation where the connection between NEHU and Utkal University will be continued. So once again, I would like to thank the department which has brought me to do work on this particular group of organisms, that is the amphibians. All right, so when I joined as a faculty there, my teacher from there, they said, why not take it up, this work, to carry on the work of my late teacher, Professor M.K. Khare. So then I had to focus my work on these little group of animals, which is a very important part of organisms, which they are the ones which have a biphasic life cycle, and they are the ones who will start then to transitional period where they will come and live in the terrestrial environment. So I will talk today mainly on the breeding biology of some amphibian species that is in our place, because it is a hilly terrain in Meghalaya. So we are at a very high altitude, and also it covers the area in the border area, the low altitude. So we find a number of amphibians which are present. If you look at this picture, this is the picture of the site of study of that area. So this you will find in the hilly terrain. We have a number of habitats which serves as a good breeding grounds of many amphibians. And when we go a little bit down in the altitude below, we find it is a plain region, more or less like what we find here. So then when we look into with our students regarding the survey of amphibian fauna, we find that sometimes it is very difficult to get the species at certain level of the altitude and also at a high level altitude. And when by the time we look into the breeding at a high altitude, by the time we go to the low altitude, we don't find anything. So this creates us a little bit of surprise. When is the breeding period? Where should we go and find them? So which leads us to propose this work on the study of the amphibians at different altitude, if there is any correlation with the breeding period, which the ecological factors have an impact on them. So now, when we look into the amphibians, the currently living amphibians, for those who are not doing work on amphibians. So just a little bit of, to refresh your memory, to look into it, we have 3 orders, the living orders, the Chordata, which include the salamanders, the anurans, that is the frogs and the toads, and the gymnophoriona, that is the limbless amphibians. Now, when we look a little bit into the history of this, regarding the number of species worldwide, this we have got from the amphibian web, up to the 20th of February when we checked in, it was 8579 number of species. In the Indian subcontinents, 446, and Northeast India, according to the paper which came in 2009, it was only 105. So far, we have not seen any update on that. And Meghalaya in the year 2021, with some Dr. Tron, when we did, he recorded it up to 61. Now, when we look into the place where this study was conducted, it is in the northeastern part of India, that is Meghalaya, in the southern slope. Now, this area where we are conducted this study, it comes under the Indo-Myanmar biodiversity hotspot, and it is marked. I have put here these pictures with many hills, then rivers, streams, drainages, and one very important that we are selecting this area, it receives very high rainfall. And then you will find that regarding the distribution and diversity of these anurans along these altitudinal gradient range in the southern part here, you will find it in India, it is available, but regarding our place here in this region, scarcity of information is there. So, the survey, the objective of this study, we have classified it into 2 aspects. One, first of all, we would like to first have a knowledge or an information on the documentation of what are the anuran species which are present in these high regions in the hills of this particular area, which receive very high rainfall in a year. And after then that, in this region, we also survey and document the low-lying area in the plain region near the border of Bangladesh. So, when after survey the altitude, the high altitude and the low altitude, then we select from those species to investigate, not all because we cannot cover, because sometimes they breed at the same time. So what I will present here is only some of those which we have managed to do. So we have studied the breeding biology of some anurans, I would say we have selected, high and low altitude water bodies of Meghalaya. Now, so the distribution is greatly influenced again by several factors according to literature survey, and also at different altitudinal gradients, it's also considered as the prime factors that influence the amphibian diversity. Already all of you know that. Now these are the pictures which we have taken in our place of study. That the anurans, they breed in diverse aquatic habitats, like you will find some of the fish ponds and then in rock pools, in rivers or streams on the sides, in the rain-fed pools, in some temporary pools, and even in the agricultural fields. I think that is a very common site in which all of you in different places, you will see this. And already we know that most, I will not say all, most anurans, they have a biphasic life cycle, that they have spend their life, the larval period in the water, and of the adult in the terrestrial. And some of them, or the amphibians, they have direct development. Now, why do we want to study the amphibians? Some people say when you are walking on board, some different groups, people, they are interested in different groups. Why we will focus on amphibians? What is the importance of amphibians in the ecosystem? Now, the amphibians, they serve as an energy-efficient trophic link. If as a students from school level we studied, we understand that they serve as a trophic link between the insects and the other vertebrates, since most of these adult annuals, they feed on invertebrates and they are recognized also at the same time as the potential indicators of the environmental health of the ecosystem. Now, recently in the year 2018, in a paper, it appears that the amphibian services to ecosystem, they are divided into 4 categories as provisioning, regulating, cultural, and supporting services. But at the same time, also nowadays, those who are working as amphibians, you will find that they have seen that there is a lot of information regarding that there is a decline in the amphibian population, and also it might be due to many climate change. It might also have an impact on them. Now, currently, there is also a massive decline, which we have seen according to the IUCN 2016. Here you will find sometimes there is a roadkill. They die due to pollution, due to predator attacks, landfill or habitat fragmentation, the cleaning of the forest. Some of them due to fungal infections, and sometimes you will find even pond desiccation. This which might be also related to the climate change. Now, so I will come to the first part. Before we go into the breeding, biology and development, I would like to go into the first part where we have to survey and document the work. So now, again, we have selected in those hilly areas. I'll go back to the diagram. So in the hill region, in these area where it is about 900 to 1,500 feet above sea level, we have selected 5 study sites or the habitats. Now these include rain-fed pools and then temporary pools, fish pond, and also the stream. We designate them as site 1, 2, 3, 4, and 5 in the high altitude region, that is 900 to 1,500 meters. Now, at the low altitude, that is in the range, we have designated again, that is site 6, 7, 8, 9, and 10. That is, it range between 100 to 900 meter above sea level. So these are the sites in which we have selected first how to document the amphibians which are present in these regions. Now, these are some of the instruments we are using, the GPS and then the camera, the dial caliper for measuring, and then the portable digital zoom, which we require for the taking the water analysis. So, and then we have also a sheet which we document the place or recording during the time of field collection. Now, coming to the results. So what we have seen, now we have taken here 5 study sites at the high altitude, 5 study sites at the low altitude, and recorded how many number of species at different altitudes. So the total number of species recorded during this 3-year period of studies was altogether in this region 30 number of species. Now, and each of the study sites, here it denotes the number of species which are present in each of the study sites collected. Now, the maximum during the time of collection of the 3 period of studies was only 40 numbers, that is the highest, and the lowest, that was 4. Now, altogether, we recorded there are altogether 7 families which have been recorded from this region. Now, when we plot, because we want to see how many are in the low altitude, how many documented from high altitude. More number are there in the low altitude, with 27 and 21 in the high altitude. So this, it can be comprehended that species richness of amphibians is found to decline with increase in altitude, as we have seen also in some of the literature. Now here this table, it shows that but there are some species which are common both at the low altitude and the high altitude. And there are also some species, 3 species, which we get only at the high altitude, and there are 8 species which are present, 9 species which are present at the low altitude, 18 which are present both at the high altitude and the low altitude. So looking at this picture, it also gives us, just the table that some of the species, why they are restricted only up to the high altitude level, and what about these which are only at the low altitude, and how come these they can adjust both at the low and the high altitude. Now, here you will find that there are 3 only at the high altitude. Now, 9 species would be at the low altitude, and the rest 18 species at both the high and the low altitude. I cannot change the slide. Sorry. I cannot change the slide. Can we? I think we can change one minute. No, next. Yeah. I think I change one. And it's not. It got stuck. All right. So now we come to the breeding biology of selected anurans, which we will study today. We will look today what is the result. So we will look into the reproductive mechanism of these amphibians, and when we look into what are the things when we look into the breeding biology, we have to look into the breeding sites, where they breed, what is their mode of reproduction, how do they make their advertisement call. Then, if during that time before the breeding takes place, if there is any male aggregation at the site of the breeding habitat, then how do they unflag, how do they spawn, where do they oviposit their eggs, if there is enough parental care taken care by these amphibians, different species, and how they are related to select environmental or the ecological factors. Now, when we look into this here, I will just give you a short brief account here. You will find that when we studied some of the species, we find that during the breeding, there is always a male aggregation. A number of males come to the pond and may be aggressive. There is a fight for the mate, and also in some, there is no male aggregation. Now, and you will find some of them in their place where they take care. How do they take care after they oviposit their eggs? Now, how do they unflag? And how do they spawn? Where is the place that they lay or taking their eggs? Different places where they will oviposit their eggs. So it shows different, in which we will come now to the next slide. Now, so for this one, we have selected to study the breeding biology complete both at the high altitude and at the low altitude. 4 species we have selected. One which is commonly seen is Hyla annectans. This is the picture. The second one is Pejobaria nepalensis, which is quite common in this area. Then Dataphrynus melanostictus, which here also you get very common, this one. And one very particular one, which we have taken a bit unique, this one, Curisalis naso. Now, what we have hypothesized also, we find that in the breeding might show a difference in the appearance, in the timing or period of breeding. So what we have seen in brief, I will show here. With this species, Hyla annectans, we find that they breed in the high altitude starting from April to May, while at the low altitude it is a little bit early, that is from the month of March till April. In the case of this species, Pejobaria nepalensis, it breeds during the period from April to early July, while at the low altitude, you will find they start appearing only from the month of May to late June. Now, Dr. Primus melanocystis, I think in the plains sometimes if you ask, they said, oh, we get this. We get this stored everywhere throughout the year. But in this place where we have selected, especially when they're during the rainy period, sometimes it's very difficult to see. But we get it only for a very short time for them to breed in the month at the breeding period, that is at the high altitude, only in the month of April to May, very short period. But at the low altitude in this particular place, we get in the winter, that is from December to January, which sometimes you don't get at all. Some people, they say, how can they breed in the month of in the winter months? But we have got it during this month. Then this one, Purisalix naso, it is not recorded at all till date. In the low altitude, it seems that it is adapted only at the high altitude, and it breeds for a very short breeding period in the month of February to March. Now, these are the instruments where, how do we do the survey to know the breeding period? First, we have to do the survey to look into if they hear the calling male, if we get unflecting pairs, if we get some eggs, and if we see any tadpoles. Then we use some of these environmental instruments for taking the readings of the air temperature, water temperature, and also for the morphometric measurements using the dialkali pulse. Now, so starting first with the breeding biology of this frog, which I have already mentioned to you, that it breeds both at the high altitude and at the low altitude. So for this particular study, we did at site 3 at the high altitude and site 9, and in the study site 9 at the low altitude, which I have told you that already the difference between the timing. Then we report the rainfall at the high altitude and at the low altitude, and here we find that it breeds from the month of April duration till the month of May. Then here also, now at the low altitude, you will find, like what I have mentioned earlier, that it breeds in the early part of March and lasted till the April. Now these are the recordings of the ecological parameters. Now, then here we record the whole year, but in this table I am showing you only during the breeding period of the high altitude, and also the recordings of the ecological parameters at the low altitude only during these months in which they are breeding. Now, then we take if there is any sexual dimorphism between the male and the female, so all the record is here. So here we find that the male is smaller than the female. And here you will find that when it starts the breeding period of this frog, it will appear and it will make a call, advertisement call. And the recording, the sound recording of this frog has been done, and it shows here it with the inflated vocal sacs here. And you will find it consists of a series of pulses which include about 19 to 20. And each of these pulses you will find again, it shows a number of sub-pulses. And for one particular call, it takes about 4,000 milliseconds. So here it shows this is the oscillogram of the call of this. And it shows that when the temperature drops below 15 degrees Celsius, the call we don't hear any call. So it shows here that it is dependent mainly on the temperature for making the call. Now, how these frogs, after making the call, then they amplex and they lay their eggs in the pristine. They select always very clear water attached to the vegetation, and they spread the eggs. And this one, we have taken it into the laboratory to look into the eggs and how many numbers of laboratory observation shows that they release about 13 to 17 times of eggs, and then at a time they release 12 to 14 eggs. So for one complete laying of the eggs, they take about 30 to 17 times. And you will find that in this particular frog, after they have oviposited their eggs, you will hardly find any parental care. That is, the adult frogs will be seen. Once they finish laying the eggs, you will not see them in the place anymore. Now, so now, then we have done also some correlation regarding the clutch size and the female SBL. So Pearson correlation revealed that the clutch size is significantly correlated with the female SBL. Now also we have done Pearson correlation tests to look with the environmental factors, that is atmospheric temperature, relative humidity and the rainfall. And it shows here that air temperature and relative humidity, they show positive correlation at the high altitude, no significant correlation with rainfall, while the number of collecting pairs is negatively correlated with air temperature at the low altitude and not with relative humidity and with rainfall. Then also we have taken the water temperature of these water temperature, then the pH, also dissolved oxygen and free carbon dioxide, and they are within a definitive range. So that is regarding with the breeding biology of Hyla and nectans. Coming to the next one, we selected this one. Also we get a study site, Phytoparia nepalensis, a study site 1, and the other one at the low altitude study, that is a study site 6. Now here we find again that the breeding of this at the high altitude, it is from early part of April and it lasts only till July. And then this is the same recording air temperature. And at the low altitude, we find that the early part of May, it lasted till the early part of June. So there is a slight difference of the appearance of these frogs. Now the same thing, we have done the recordings only, we have done the whole thing of the recording of the ecological parameters of the year, but here we are showing only during the breeding period of this particular species, that is at the high altitude and at the low altitude. Now here the morphometric measurements of these frogs also have been taken. Then the same thing, emergence of the male first at the reproductive site by making the advertisement call, and then after making the advertisement call, then the female will appear, and then amplexus will occur, and then after amplexus, then they will oviposit their eggs. Now what we have found, that during the period, we have brought also some of the amplecting pairs to the laboratory, and they lay eggs in the laboratory, and these are the eggs which have been recorded. Now then we look into also the same thing, the amplecting pairs. The clutch size range from about 301 to 443. Now when we look into the Pearson correlation, it reveals that the clutch is significantly related with the male SBL and the female SBL, and also there is a positive correlation between the male and the female SBL. Now then we look into the air temperature, relative humidity, average daily rainfall of the study sites. That is, this is at a high altitude, this is at the low altitude. And we find that with the air temperature, there is a significant correlation, but not with relative humidity between the high and low altitude, and also with average daily rainfall, there is not much of the maybe like correlation, but and it shows it is not significant. Now, then Pearson correlation between the air temperature and the number of collecting pairs and relative humidity was seen at the study site at 1, that is at a high altitude. No positive correlation with rainfall, and at study site 6 with the green dots, here you will find that is the number of amplecting pairs, it correlates only with relative humidity and not with temperature and rainfall. Similarly, the water temperature of the study site, it is within a definitive range during the breeding period. Now, coming to the frog, that is Dotaphrynus melanostichus, which is very common, that you get all over here, I mean, and even in our place, we get in this place that is both at a high and the low altitude. Now, but interestingly, we find this particular frog in the, it breeds at high altitude. The onset of breeding of this toad, it is during the monsoon, and it lasts only for 2 months. We have surveyed this area so many times, and only during the month of April to May, very short period. And with the study of it, at the peak, you will find, while at the study site of low altitude, we find that the peak of the breeding activity, it is in the month of January. That is in the winter, when the temperature also more or less high and low altitude, it is similar. So this is the same, we have done the ecological factors, the same thing. So the pattern of emergence of this at the breeding site, it is rarely observed. We'll find it will make some advertisement call. It will mean while calling, the male will show very less inflated vocal sac, and throughout the survey it revealed that no unflecting pairs were seen during the night. So here you will find again, if the temperature is below 15 degrees temperature, you will find that it shows that there is no advertisement call. So again, here it shows that it is dependent on temperature. Now here is what we observe, the amplexus. Mainly it takes place in the daytime between 11 to 14 hours. We have been in the field, and it shows here how it lays its egg, and it takes many hours, and the frog will move slowly in some straight or sometimes curve, and it will attach these string of eggs in the vegetation. Now we have done the morphometric measurements here regarding the male and the female. Now here we have done also the amplexing pair of males and females on the basis of their body size. It is commonly observed that it's the large males, they pair mainly with the large females. Now here it shows a correlation between the male and the female SBN. Now this one, it shows the clutch size with the female and the male SBL. So here they are about the clutch size. It is very large. It's very difficult. This is approximately what has been taken counting by measuring only how many in one particular string, which is a measurement of how many eggs will be there. So just multiply that, because very long strings of these eggs we're getting. Then we have taken also the air temperature and the relative humidity of both the study sites at high and low altitude. It shows it's not significant even with relative humidity. Then this is the Pearson correlation of the positive correlation between the air temperature and relative humidity at high altitude, and with the number of unflecting pairs, and but no positive correlation in case between the rainfall. And in winter, there is no rainfall during the month of January. Regarding the range of water, it is within a definitive range, and these are most of the places not much disturbed in these rural areas. Now, coming to this is a little bit unique frog in which we have located only at high altitude. That one is a very small tree frog, Puricalax naso. From the month of September to January, this is the area where we have located about 6 meter height. They hide between the tree holes in rock burrows, even in small rocks below it. They will rock crevices. You will find they are present during the hibernating period. Then, with the coming of the rain, shower of rain, the first shower of rain, you will find that these frogs, they will start to appear. And then at that time, the soil temperature, the soil moisture content will increase and the temperature will decrease. So we have recorded the temperature, the soil temperature, soil moisture, and the air temperature and relative humidity. So with the first shower of rain, then they start appearing, the unflecting pairs will come, and they will make a very faint fall. And then after they make a very faint fall for continuously 7 to 11 days, then the female will start appearing. So when it will start appearing, then they will unplug, and then they will, before they will unplug, they will make the advertisement call. This is the advertisement called the oscillogram, which consists of just 3 pulses, and some of these are small sub pulses. And then you will find that between the duration between here and the second pulse, it is different and then a little bit irregular, we would say. So then after that, then they will go into the burrows, inside the burrows, and they will lay the eggs. And after they ovipositing the eggs, they will oviposit the eggs inside the burrows, and the male will kick the eggs out from the burrows outside. And a very nice observation, when we went to the field, we found that these eggs, they mix with the soil. That if you are not careful, if you are not aware of these things, you will not be able to see. The first time when we went to the field, it was amazing when we see. Then they appear like this. You cannot imagine that the eggs, they are laid in the soil and they are mixed with the soil, that exactly, it's not in the water, that they look exactly like some small seeds of like small big stones, but very fine, very nicely covered. That even if you are not aware, really no one will, they will stand and walk straight on. And amazing things of these things, they will last after they oviposit like that, it camouflage with the background. So then you will find that after a few days, they are laid like this. When the first shower of rain comes, the egg jelly which covers these eggs, it will burst. And the tiny hatchlings will come out, and immediately at the time it will be followed with the rain, and it will be filled, these small ephemeral pools, very shallow, and then they will come out the small tadpoles. And then later they will fill in with the water, and they will start developing, and they will pour down these tadpoles to the bottom. That is, they are benthic organism tadpoles that they feed below. You will not be able to see. So this is one amazing, because it is a rhacophorid, in which mainly, I will come to the next slide, they lay mainly eggs, foam nests. But very amazing thing is this one, it does not lay a foam nest. I will come to the next slide. So this is the blood size of this. It is only about 63 to 120 eggs. So there is a positive relation between the male SBL and the blood size. But again, another thing, there is no correlation between the blood size and the female. So here is that maybe the male the SBL and the blood size in Nassau, it is rarely observed in other amphibians. And here you will find it with what we suggest, like some other workers, that maybe the larger males can fertilize higher proportion of the eggs in the flocks. So here is the eggs which we can see in these very prominent, that you cannot imagine that they are amphibian eggs. Now I will not go into the detail how the process of breeding biology goes on. Now this is another polypidis. I'll just tell the duration of the time. In the high altitude, we find them in the month of March to June, and then they make the foam nest. It is in this way, in the grassy vegetation, and then which is a little bit moist, and they make foam nests. Another species which is a rhacophorid, it is Polypidis. In the high altitude, we did not record the breeding, but only at the low altitude, it is from the month of March to June. While they also lay near where water bodies are there, they will, in a moist, with a little bit submerged in the water, you will find they lay eggs in the foam nest. Now, again, here you will find this as a very common. We have worked the detailed work on the breeding biology of this frog, Rhacophoros maximus. There is so much male aggregation fight between them, between the males, and then they lay eggs mainly near the substratum, in the terrestrial region, and also in the grassy vegetation. Now, this is a Rhacophoros bipunctatus. It records its high altitude in the month of April to July. You will find also, even on the tree trunk, in the leaf, they lay their foam nest, and just below it there will be some water. So when they come out, the tadpoles will just drop, hatchlings will just drop in the water. So here just it will be very near, close by to the water body. So here we will find that the reproductive modes exhibited by these species which we have studied, like Rhacophoros the first Maximus, it shows the different mode, that is mode 11, 28, 30. There are explanations what are. When 11 mode, it shows the foam nest floating on the pond, and then mode 28 foam nest on the human forest floor, and mode 13 foam nest in the subterranean constructed nest. Now by punctators, it shows mode 24 and mode 28, that X on the leaves and then foam nest on the human forest floor. Now Himalayensis polypodids, it shows mode 28 foam nest mainly on the human forest floor. Now the other one, Polypodids terraensis, you will find it shows mode 28, that is on the forest floor. By Pudicalax naso, it shows two modes, that is X and early tadpoles, they are inside the burrows. Sometimes when they don't find burrows, they cannot find burrows, you can find them even on the open soil surface or above the rocks, but on hatching, that is only when there is some rain, shower of rain, that is done. This is one thing which it has created an interest in us. If there is no rainfall, then all these eggs which are laid, they will die due to desiccation. So this is one in which further work can be conducted on this. Regarding Dotaphrynus, it shows mode 1, and then also Hyla and Ectins mode 1, the egg and the exotrophic tadpoles in the lentic ecosystem. Now, so when we look into the timing of emergence of these frogs which are present in the both at the high and low altitude and those which are at the low altitude, we find there is a difference in the timing of emergence and the duration of the breeding period. So like Nephalensis, that is at the low altitude, it shows a little bit shorter duration than at a high altitude. Then regarding again Hyla and Ectins, it appears first at the low altitude, then later at the high altitude, and the duration is a bit longer. Now coming to Dotaphrynus, it is in the middle of the year, that is April to May, but at the low altitude, you will find it starts in the month of December and continue till January of the next year. Then Polypodids, Himalayensis, here you will find it is more in the low altitude, and then later at the low altitude it appears, but it is in the high altitude, it is for a very long time, the duration. It appears a little bit early and also take a longer time, the duration period. Here now, so very short period and only for 2 months, where bipunctators we have recorded here only at the high altitude. Now here you will find also it has a very long period, and similarly Maximus, it is found both high and low, but regarding its time during the study period of these 2 years, we have managed only to look at the altitude, the high altitude regarding its breeding period. So with this particular study, we have seen that we have come across that due with the use of the in the sound recorder, we find there is the difference in the call between the different species. That is, different species, they have a specific call for a particular species. Now, and one important aspect is that during this study, we have come across that TIASRST, which shows an interesting terrestrial oviposition in which it does not lay eggs in the water but in the soil. Now, it was found that breeding activity, they are commonly found in both high and low altitude, like these species which I have mentioned, but the timing of emergence and the breeding period shows slight difference. So this studies that the combination of might be of many ecological factors, which some we have not taken into this particular study, might play an important role. So in most cases, we find that the role of rainfall acts as a primary extrinsic factor governing the breeding activity. But in some, we find it is not, because in the in the case of Datifrinus, in the low altitude, when there is still no rainfall, still it breeds when at the time of its breeding period. So the study, it shows here that it harbors a number of amphibian species. Now we have got only we have got nearly 7 types of reproductive modes from just 8 annuran species. So here it shows the rich diversity of the amphibians in this particular area. So with this, I would like to conclude my talk on this aspect, and I would like to thank the Department of Zoology, NEHU, for providing us all the facilities for conducting the work, the local headmen of that particular area where we have done to venture into the forest, the fish ponds, and all the places with their permission. And also the study was supported by the STSRB New Delhi. And then I would like to thank my scholars, that is Dr. P. Shankliang and R. Shadab, who have done a lot of work on this particular aspect. Thank you.
So it's a very nice talk, very important. So we will take 1 or 2 questions.
Dr. Rupal, wonderful talk. I really enjoyed listening to all the work that you're doing, particularly with the NASO, curriculum NASO terrestrial egg-laying behavior. I think it needs a lot more investigation in terms of what substrates are being used by the animal, and that might be very crucial. Even the microbes associated with the soil, which might protect the eggs. And what stage of the tadpole hatches out from the egg? Can you tell me that?
Yes, that is very interesting, because in other frogs, we find at stage 22, meaning they hatch. All right. But this particular frog, when we observe, because we find that with the coming of the rain, then it bursts straight away. When it bursts, it already reached stage 25. All right. So then, because when we find other frogs by the time of stage 25, it will take from stage 22 when they hatch, it will take some time. But it is amazing in which this time we thought that immediately when we go back from here, because this is the timing, to do a little bit of further analysis regarding the covering. You know, when you look at the cover, it all mixed with the soil cover. We don't see anything. Then myself, Dr. Pankit, and Mr. Ricardo, we were trying to open it. It is very tough, the jelly which covers. So we thought further we will do work on this. So that is what because this it might shows, like what I've shown, other Rhacophoras, they leave foamness, all right, with all the thick gel in the cover with the white foam. But this one, outside in the soil. And we have observed, I think in 1 year, before we started this work in 2016, when we went to the field with my students, when we look at the field, we find that it is spreaded, and all these, I think, have been trampled by dogs and stuff because it was open. So maybe it might require a little bit of what I would say, might look at some endangered species also, because we don't know. If the rain doesn't come, also they will disappear. So anything suggestion besides that, what is we should do with this? Then I would say one thing, what I feel, it might be one step ahead which with the amphibians will move towards terrestriality. I don't know, that is what is or any more questions? So before I will I would like to thank the organizer, and particularly what I've expressed from the first time of my talk of Professor Datta is here now, that I owe my gratitude to the Department of Zoology Utkal University for motivating me late back in 1990s to take up the work in amphibian science.
Very nice job. Very thank you.
Thank you, madam. Now time we need to felicitate you, for which may I request our chairperson, Professor Pal, to felicitate Professor Hulur. So, please. So there is an announcement. 20 posters are to be presented. We will start the poster presentation at 5 p.m. And parallel we'll have oral presentations. So over to Professor Pal.
So we'll start now the oral presentation. So we'll have 10 minutes presentation, 7 minutes plus 3 minutes. So we like to request all the students to stick to the time so that we will move with this session.
Now, may I please invite on stage Ms. Kalika Patnaik for her oral presentation.
No, really. Hello.
A very good afternoon to all of you. First of all, I would thank the organizers for giving an opportunity to present my work here. So I will be talking on the vermicomposting, which will be an effective bioremediation for solid waste pollution by soil sanitary napkins. Now we know that the soil microbial ecosystems, they play an important role in maintaining the nutrient pool through mineralization of the organic matter, and the soil microbial biomass is one of the agents of transformation, and on the rate of decomposition, it depends mainly on three major factors, that is the soil organisms involved, the physical environment, and the quality of the organic matter. And the organic matter on the basis of the rate of decomposition can be classified into three groups. One which rapidly decomposes. The second one is the hemicellulose and cellulose, which are less rapidly decompose and very slowly, like fats, wax, and waxes and resin and lignin. Now these are the important enzymes which are involved in the composting process. And I will be talking on the cotton decomposition because this is much important for present scenario. In India, estimates say that 26 lakh tons per year is produced, and out of this, and if we think of the sanitary pads used by an average woman during her menstruation, per month uses around 10. So if you can think of in a span of about 35 to 40 menstruation years, she accumulates about 125 kilos of soiled pads. And in India, there are about 355 million women of reproductive age. So you think we can imagine how what a huge amount of this soiled sanitary feminine napkins are produced during a year. Now it not only pollutes the environment, atmosphere, it leads to infectious diseases, and this particular waste, it is disposed of by burning, and once it is burned, then again it increases the carbon dioxide level. And the most way of this particular it is going to the landfills. So therefore it will be creating problems by anaerobic decomposition of this decomposed matter. Now what we told of that this particular waste can be collected, processed and converted into biocompost by use of composting and vermicomposting, that is the use of the earth. So the present study, the attempt was made to analyze the process of decomposition of the soil waste during using amendments and earthworms through vermicomposting. So basically the material for the particular experimental setup was divided into 4 groups. The first group consisted only of cotton and soil and unused cotton. And then the second group of the 1 kilogram of the soil with which we use cotton. Then the third group, we made an amendment of cow dung so that the microbial decomposition is facilitated. And the fourth group is the vermicomposting group where the earthworms were taken along with the cow dung and the used fat. So the different the standard methods were used to study the enzymatic activity, and the research what we found that protease activity, which can be utilized or considered as an indicator of decomposition, we found that it was highest in the vermicomposting group. So similarly, when we thought of this urease activity, there also highest activity was found by 60 days in the vermicomposting group. Amylase activity was also highest in the vermicomposting group. Cellulose activity, most important, that is the degraded cellulose takes very long time for degradation, and there also that was found highest in the vermicomposting group. So from this study, we can conclude that enzymatic activity can be considered as the driver of this biological stability of the organic waste, and the solid waste, which takes longer times for decomposing itself, can be processed through the vermicomposting, and by the process, we will be converting waste into an organic fertilizer, that is the vermicompost. Thank you. And the earthworms which we used were the local species which are present in the locality of our university. And basically the epigenic and the anesic earthworms were utilized for the vermicomposting. Thank you.
Okay, so 1 or 2 questions.
So may I request our chairperson to present the certificate of participation, Tina ma'am. Thank you.
Now may I please invite on stage Vinita Pandey ma'am for her oral presentation. Be on virtual mode. Okay, due to technical difficulties, we will move on to the third oral presentation. May I please invite on stage Sunanda Sahu ma'am.
Okay, so very good afternoon to one and all present here. Myself, Dr. Sunanda Sahu, Assistant Professor Stage 3 in School of Life Sciences of Bangalore University, and going to present a study on potentiality of fish and old termitarium soil and biofertilizer. I think it is surprising for inhabitant of the public because it is termite is a nuisance creator in the particularly in the building and all. So now coming to my contents, as we all know, there is environmental degradation due to the uncontrolled use of chemical fertilizers in agriculture, and that has actually forced several environmental scientists to find an eco-friendly way to grow crops using the biofertilizers, which can improve the soil without compromising the soil. And as we all know, termites, they are with the ecosystem engineer. I can say it is one part of the coin, another part definitely it is a nuisance creature causing so much of harm. But on the other side, it is a good ecosystem engineer modifying the soil condition and creating rapid turnover of these linear materials so that it can be decomposed easily. So then termites, they are considered as important component of our ecosystem, particularly in developing economies where they can alternatively they can be used as an agri. So importance, already I have told that they improve soil fertility and create microhabitats that can regulate the structure of soil, bacterial and fungal communities. And also there are fungal garden and fungal combs which are present in the subterranean termites that help in digestion of the cellulitic and lectocellularic material which are very hard to decompose. So also they are very helpful for improving water filtration, nutrient leaching, and surface runoff and drainage. So coming to the objective, as we know, despite their well-known role in controlling carbon and nitrogen fluxes in tropics, but termites attracted less attention of researchers, particularly in India. Keeping this in view, we have studied a study on potentiality of fish in old termitarium soil as biofertilizer, taking these parameters into study. And before this, so we have published one paper in Journal of all these things. So now we are studying, we are studying this aspect of the termites. So coming to the study side, we can see here, this is the Sambalpur University campus. There is a hill inside the campus that is known as Dehraduni, and towards the Hirakud Dam, there is a hill known as Dangri, where we have collected the old and the fresh Termitaria soil, Termitaria soil. Then next these are, so CFM translating the fresh mount. This is there is only inside campus. Fresh mount, this is old mount, that is. And how we know the mount dates which are collected from different sites, from November, we have collected it and we have processed this Termitaria soil in the land. Then next, how we identify is the fresh and old Termitaria soil. That the fresh mount was identified as patches of moist, very raw, small conical deposits on the surface, whereas the old mounts, they are very well consolidated and to form a conical shape and with a variable height. And this, the nutrient content was analyzed for TIASRST protocol, and later on we conducted statistical analysis, one was followed by 50 pairwise comparison to using Minita 1870. Then coming to result and discussion, I am thankful because we have taken the termites we got from this site to identify Kolkata for identification, and after the observation we find these two species, this is the Odontotermis officers from Dehraduni inside our campus, and Odontotermis T from Chandri, that is near the Hirakud Dam. Then we have studied textural composition, that is significant difference in stage in the old Termitaria soil, where we can see in the old, particularly in the fresh nest, there is clay content is significantly less than the old nest soil, because they have consolidated structure features in there that accumulate a lot of clay materials in this. And regarding soil pH, the soil pH range from around 6.9 to 5.83, which can which would meet most of the pH requirement for crops if you can use it as an amendment. Then the better amount of soil pH in Mount soil, particularly in Dehraduni, in particular in our campus, particularly that may be due to this better clay accumulation and better rate of matter decomposition by termites. But however, no significant difference was observed here. However, when we consider soil conductivity, that is an index for soil water soluble salt, we can find that greater level of fine sediments and soil organic matter in dormant mounds, particularly in this Dehraduni area, will that is cause higher electrical conductivity. And as I am talking about its implications in the agricultural context, that's why I have referred that by agricultural point of view, soil with electrical conductivity greater than 4 decibels per meter are considered saline and not suitable for paddy crop. And that we can observe all these that below the 4 decibels per meter. So, in from agricultural point of view, we can use the poor farmers can use it in their crop. So, then coming to organic carbon, how we find that organic carbon, so it is less from the surrounding soil, of course, we have taken the soil, and particularly in old and in fresh, there are less organic carbon can be due to the high active. When they prepare the mound, they are very highly active, and this dot microbial process of termites and its specific activity is high, so leading to more consumption of carbon. So, and coming to the nitrogen content of termite nest, it ranges from 1.45 to 1.73 gram per cent, with significant difference between fresh and old mound soil. And this high nitrogen in fresh mound in comparison to old might be contributed by combined action of foraging and enzyme activity by residing in the mound to convert this material to available nutrition. And as I referred, and in specific Indian paddy variety, the low depth speaker relatively low nitrogen. So, and this coming under yes, time over. Okay, just so significant difference we can have with phosphorus and potassium. We can say definitely as well as potassium in test form cell and surrounding cell would begin to the decomposition of organic matter and concentration of greater amount of clay content with genes by the termite. So, and in that observation, we have done by Anupa and post of days, and so then I just I refer to another pattern, the fertilizer tools, and I found that the observation we made from this study that the poor farmers in this area, which they have abandoned the field, they can use this for their profits. So in conclusion, I can say from this story that emphasizes the use of mixed traits and one-dimensional soil by small stakeholders or small farmers and small farmers and we can mix them. But however, further intensive study in this context is recommended to arrive at complete conclusion. So I acknowledge our head of the School of Science for the providing facility, and also I am very much thankful to JSA India for identifying the termite species. And finally, references are there, and thank you for your
Thank you, ma'am. Can we have one or two questions?
So, yes, there is no question. So everybody, I can understand everything. So thank you.
Ma'am, we have lot of termite in our campus, so.
Here, method of work is done on the aspect of how method of insecticide may fail. But I am focusing on the other part of the point, that is their positive role. Even if I have had my house in government, lot of termite treatments are there. I have done that, but at the same time, everybody has a positive part and negative part. So,
May I request now our chairperson to hand over the certificate to Dr. Sam.
yes, thank you.
Now we'll have the virtual mode oral presentation. Now may I please request Vinita Pandya ma'am.
National Scientific Council on the Developing Child has clearly stated the beliefs from different parts of India. My top respected chairperson and my other colleagues from different parts of India. Uh, my topic of today's presentation is epigenetics, social determinants and transformation, nature and nature. We are seeing that there is emerging scientific evidence that whatever is the internal body conditions, it is just not influenced by the genes, but also the various other associated factors. In fact, National Scientific Council on the Developing Child has clearly stated that experiences being a chemical signature on genes that determines whether and how genes are expressed. Genes are not set in stone. That is to say that genes are not the only determining factors in development. In fact, large body of literature, scientific literature and evidence is also suggesting that epigenetic mechanisms, which are molecular modifications that is regulating gene activity without changing the DNA sequence. Though I am a student of psychology and I do not have a very thorough understanding of the genetic functioning and structure, in recent times, a lot of technological innovations and scientific research on epigenetics has emerged, and Bruce Lipton, the very strongest proponents of his writings, his research is truly inspiring. So when we talk about epigenetics, it is a study of how behaviors and environment can cause changes that affect the way genes work. Unlike the other genetic changes, epigenetic changes are reversible and do not change the DNA sequence, but they can change how our body reads the DNA. And social epigenetics is also strongly emerging and researched by sociologists, psychologists, bioethicists, and professionals related to public health. They are also trying to explore that how social factors determine the gene expression and can transform them. When we talk about genes and epigenetics or epigenome, I'm reminded of Dana Dolino and Environmental Health Sciences and Nutritional Sciences at the School of Public Health University of Michigan. Dana says that genes is like body's hardware, epigenome is like the software. So almost each of the roughly 250 different cell types in the human body has a different epigenome. And without changing the genetic sequence of DNA, those epigenomes activate and suppress genes through DNA methylation and histone modifications done through the genetic processes. So, in a layman's language, for people like me who are not from hardcore science background, to make it very simple, Dana very beautifully says that epigenome acts like a sort of a dimmer switch that can promote a gene to turn on slightly or turn on significantly or turn off. That is the power of the epigenetic influence. And genes are hard coded at conception. The epigenome is dynamic and changes throughout the prenatal development and life. If we have to quote our researchers like Girolo and Atkins, who also have come up with very interesting findings that social epigenomics is a new field of research that studies how social environment shapes the epigenome and how in turn epigenome modulates behavior. So there is a very clear implication and correlation and lot of sociological studies that how social adversities can lead to greater negative health outcomes. Such kind of research is strongly emerging, dear friends. And there are several social determinants and factors which is changing epigenetic mechanism right from our diet, obesity, to stress, to medication, to hormones, nutrition, development, and aging. Similarly, epigenomic changes the
impact, and it can have tremendous impact on our brain functioning, and our health can trigger conditions like cancer, diabetes, autoimmune diseases, mental health problems. So apart from the environmental exposure, stress is also an important component, social component, which influences epidemic functioning. So, to quote a very wonderful study by Jennifer Smith, who is professor of epidemiology and human public health, has studied that the impact of neighborhood and social environment on genes related to stress and inflammation. In one study, she and her colleagues found a correlation between poor neighborhood conditions such as safety concern, low social cohesion among neighbors, and poor aesthetic quality and DNA methylation in 18 genes related to stress reactivity and inflammation. So other qualities of neighborhood, including a lack of food availability or exposure to environmental toxins, could also influence methylation of genes and other genes as well. There is a very strong relation between epigenetics on how it is mediating the outcomes such as depression, anxiety, schizophrenia, and addiction. And the current mental health epigenetics supports that adverse psychosocial experiences are associated with mental health disorders such as schizophrenia and addiction. The core of my presentation is that there is a very, very strong reference of epigenetics in our Indian Ayurvedic system, and the correlation basically emphasizes on lifestyle information because Ayurveda plays tremendous importance to environment, including person's behavior and social environment. And that is why our ancient take on epigenetics very clearly says that what you see you become. So, development of epigenetic research integrates the social as a positive factor for biology of disease such as we rarely see in the other kinds of genetic research. And it is estimated that 90% of life is caused by epigenetics changes in gene expression brought about by what one does in one's life. So, whatever is done, the phenotype or a Ayurvedic psychophysiological constitutions in our daily prakriti is laid back to the DNA and changes its expression accordingly. That is to communicate in a very simple languages, our action or our karma impacts at the cellular level. So if our activity is, it includes your fantastic breathing exercises like the Kapalbhati, Anulom Vilom. That kind of breathing activity will also influence our functioning of the cell. So indeed, very strong correlation about epigenetics and its influences defined in our Ayurvedic system. So social determinants are definitely influencing the internal environment of the body, and there is potential to bring its transformation as well. Genetics and epigenetics are very clear-cut reflections of nature and nurture, and we have seen that ancient Indian medicine and wisdom indeed have clear references to strong correlation nature and nurture, and Ayurveda very strongly clearly establishes the epigenetic influences. The technological innovations through epigenetics is definitely a watershed development in our scientific research. The contemporary epigenetics research is still correlation with genetic experts. There is more need to have more conclusive evidence to research that epigenetics is impacting human living, but whatever scientific research has emerged still now, those evidences are indicating that epigenetics is influenced by social determinants and can bring social transformation. So I thank all my friends from the university, and I strongly apologize for my absence during this very, very prestigious. Thank you all very much.
Let's invite Balay Ghosh for